So good afternoon and good evening, everyone. We want to stylishly wait for everyone to start the next 1 or 2 minutes. We want to stylishly wait for everyone to start the next 1 or 2 minutes. I'm sorry, I think I was not audible before. So I said we are looking at classification of bacteria into different groups based on their similarities in skin, such as phenotypic similarities. I think I was not audible before. Physiological. So I said similarities. We are looking at evolutionary relationships and so on, based on their now similarities. This classification of bacteria serves various functions which are used in grouping them based on different typing schemes. Physiological. We shall be looking at them one after the other. I hope you can hear me loud and clear because it seems some of these audios are having some backfiring. If you can hear me, please type yes if you can hear me. So that I can continue. All right. Can you hear me? If you can hear me, can we continue? So, talking about classification of bacteria using different schemes, we want to look at these schemes one by one. We have morphological characteristics, we have growth characteristics, we have antigenic properties, biochemical characteristics, and pathogenicity and toxin productions. So for morphological characteristics in bacteria, these are observable characteristics that are found among microorganisms. Example includes how these organisms have shapes, appearances, and so on and so forth. So for morphology and morphology characteristics, this includes the shapes of these organisms, the arrangement of the organisms, that is the bacteria, how they are shaped, how their cells are shaped, the arrangement of their cells in clusters. For instance, we have the cocci, we have the rod shapes, we have those that have curved shapes. Microorganisms are based, they are grouped based on these shapes and sizes. And also how they appear under the microscope after gram staining is also very important because it allows us to know gram-positive bacteria and gram-negative bacteria after they have been stained under the microscope. Some of them have flagella, some do not have flagella. So we can also classify them on a scheme of motile bacteria and non-motile bacteria. Again, we have those that have spores, sporulating bacteria. We call them sporulating bacteria because these ones, they can produce spores. And those that have capsules that are shielded. We talked about capsules and spores previously in our last class. So there are different staining methods that helps in classifying bacteria. Another is acid-fast stain that are of importance in classifying Mycobacterium tuberculosis. So the information on morphology and staining reaction of an organism can help us to identify them up to the genus level. And it can help to minimize the possibility that they belong to another group of organisms which are not of the same shape or not of the same size or not of the same cell arrangement, especially during classification. I would like to know if I'm very audible. Let me stop sharing this screen so that I can check if I am audible. If you can hear me loud and clear, kindly, okay, I'm audible. Thank you very much. Because the network sometimes could be funny. I don't just want to continue talking while you are not listening to me. So I will resume back in sharing my screen. Another scheme is their growth characteristics. Growth characteristics for microorganisms, it is how they appear on their colony, the colony on solid media. How they appear on solid media. Different microorganisms have different colony formation. Some of them produce pigments. Some, for instance, Staphylococcus aureus, if you culture these bacteria on specific agar, such as mannitol salt agar, remember, Staphylococcus aureus can be isolated specifically on agar called mannitol salt agar. This mannitol salt agar can actually allow Staphylococcus aureus to grow with a characteristic golden yellow colony formation. And this formation immediately allow you to know that this is Staphylococcus aureus, other than any other bacteria that is growing. So also we have Escherichia coli that gives metallic sheen color when you grow it on a specific media called eosine methylene blue. Some of these identification process will be carried out during your practicals, and you will be able to see what I'm talking about. When you start doing the practicals, you will see some of these characteristics I'm talking about. Now, this growth characteristics for microorganisms allows us to see how they are different from one another. So it can be easily grouped with those that have similar growth appearances on solid media. One of the primary distribution characteristics for organisms is also how they grow aerobically, anaerobically, some facultatively, or microaerobically. So we will look at these two and give some definitions to them. When we talk about aerobically, those organisms that grow in the presence of oxygen, and those ones that grow in the absence of oxygen. We also have those that have adaptability for whether there is oxygen or there is no oxygen, they grow. So based on their growth characteristics and air tolerance, bacteria have been classified as obligate aerobes. Obligate aerobes require oxygen for their growth. Example includes Pseudomonas aeruginosa, Mycobacterium tuberculosis. They require oxygen for their growth. We also have obligate anaerobes. Obligate anaerobes, they do not tolerate oxygen. They could die in the presence of oxygen. Example is Bacteroides fragilis, which can be found in the guts. We also have Clostridium tetani, which can cause tetanus, a wound infection. These are obligate anaerobes. Another species of Clostridium called Clostridium botulinum is also responsible for food poisoning. So when people close their food in a tight container without allowing air to come in and without having a way of preserving this kind of food, it allows the proliferation of Clostridium. And when these organisms grow, especially botulinum or perfringens, they can easily produce toxins. And when such food is consumed, it could lead to food poisoning, in some cases, death. So we also have microaerophilic. Microaerophilic requires low oxygen concentration for growth. Example, we have Campylobacter jejuni. We also have other examples such as Helicobacter pylori. They require low oxygen, like 5% oxygen concentration. Others, they prefer more of carbon dioxide, but 5% of carbon oxygen. So we call them microaerophilic. Then we have facultative anaerobe, also known as aerotolerance. They can grow with or without oxygen. Example is E. coli, Escherichia coli, Staphylococcus aureus. These organisms are classified based on their air tolerance, how they tolerate the air tolerance, how they tolerate the air in their growth form. So that means when they are growing, the measure of air that is available to them will determine their growth condition. So you can easily classify this organism based on how they grow in the presence or absence of air, or at the moderate provision of air, which is microaerophilic. Other important assessments include the incubation temperature, pH, nutrient requirement, and resistance to antibiotics. I will explain this. Other organisms have temperature tolerance. We are still going to look at it. And there are others that also prefer some level of acidity or alkalinity. Then nutrient requirement. Some organisms can grow in minimal medium content. That is, they can grow in medium that does not have sufficient carbon sources that is required for average organism to grow. Such organisms are in the environment, they are so rugged, they are found almost everywhere. Example is Pseudomonas aeruginosa. This particular bacterium can grow inside a distilled water. A distilled water is a medium that is not expected to consist of any supporting element, nutrient for microbial growth. But inoculating Pseudomonas aeruginosa in such medium of water, there's a guarantee that Pseudomonas can grow, can remain persistent in this water because it requires just minimal nutrient to survive. Whereas there are other organisms that will not grow if there are no abundance of nutrients. We call such organisms fastidious bacteria. They are fastidious in their growth requirement. That means what they require for their growth is essential nutrients, fortified nutrients, abundant nutrients, and some enriched medium that will be supplied in abundance for them, and they will grow. Example of such microorganism is Streptococcus. Streptococcus pneumoniae, Streptococcus mutans. These organisms are fastidious in their growth requirements and also resistant to antibiotics. There are some organisms that also develop resistance to certain antibiotics that can easily kill other bacteria in the growth medium. I mean, in the medium for growth. So it serves as a basis for selecting certain bacteria while cultivating them. Because, for instance, bacteria known as Enterococcus. Enterococcus are gram-positive bacteria. I hope you are taking notes. I hope you are taking notes some of these examples I'm mentioning. Because of our time, I do not wish to put a lot of details in the slide, but I feel some of these things, when I'm mentioning it, you can jot them down. So Enterococcus is a type of gram-positive bacteria that is naturally resistant to antibiotic class called cephalosporin. I'll take that again. Enterococcus is a gram-positive bacteria that is naturally resistant to antibiotic class called cephalosporin. So cephalosporin are specific beta-lactam drugs that can inhibit both gram-positive and gram-negative bacteria. But Enterococcus have a very sufficient way of developing resistance. So because naturally it is resistant to this drug. So if Enterococcus is to be selected in a medium, if you put cephalosporin drug in such medium, it will inhibit bacteria such as Escherichia coli, E. coli. It will prevent E. coli from growing, but Enterococcus will grow. So it's a way of selecting such bacteria in the environment. So we also have an example of Campylobacter jejuni. It grows well at 42 degrees centigrade. This 42 degrees centigrade will normally inhibit the growth of bacteria such as E. coli. Because at 42 degrees centigrade, this temperature is too much. So E. coli will not grow. E. coli typically they grow within some of them are normal flora. They are found in the gut. And the normal body temperature of every human being is around 37 degrees centigrade. So any temperature above that becomes more intolerable. So E. coli will not grow at 42 degrees centigrade. Additionally, Campylobacter will grow in the presence of certain antibiotics that are used to select it from other organisms. The reason for this for Campylobacter is because Campylobacter is a slow-growing bacteria. It doesn't grow faster. So these antibiotics help to slow down other organisms, and the temperature also serves as an inhibiting factor. Another growth condition is an example of Escherichia coli. They can grow on minimal media. They grow very well on McConkey agar. Although McConkey agar consists of other inhibitory properties that prevent gram-positive bacteria from growing, but they will grow. They will grow on nutrient agar. They will grow on McConkey agar. They will grow on cleared agar. They will grow on some media that does not have. They will grow on Salinity that does not have enriched facility for the growth and cultivation of fastidious bacteria. So let's look at temperature-dependent organisms. There are organisms that they grow based on different temperature requirements. These are very popular. We have the thermophiles. Thermophiles are found in hot environments, such as hot springs. Example is Thermus aquaticus. These organisms have a way of adapting to this environment. Their cells have been adapted to withstand the high temperature. They do not have such type of lipids that can be melted by high temperature. So they have adaptive features that allow them to go well in this environment. We also have the mesophiles. Mesophiles are the abundant class of microorganisms because these organisms grow at normal temperature between temperature of 20 to 25 degrees to 35 to 39 degrees. These are mesophiles, and they are mostly human pathogens. They are responsible for causing disease in both human and animal, and also, of course, in plants. So example is E. coli, we have the Klebsiella pneumoniae, we have the Pseudomonas aeruginosa, we have the Staphylococcus aureus, We have the Proteus mirabilis, we have the Enterobacter, we have the Enterococcus, and so on and so forth. So we also have the psychophiles. The psychophiles are those that grow at a lower temperature, below 16 degrees centigrade. They grow and they proliferate at a lower temperature. Most other organisms will not grow in this temperature because the temperature is too low for their metabolic activity. It freezes their cells, it freezes their lipid content, it makes them to be inactive. And this situation is not favorable for them. But psychophiles are active. Some of them can grow in the refrigerator. They have capacity to grow in the refrigerator. They spoil food. One of the significance of psychophiles is that they are food spoilage bacteria. When some food items are kept in the refrigerator and are contaminated with psychophiles, they can easily spoil food. So please take note of these. These are things that can come out in your examination, because if you are asked to name or give a class of bacteria that's actually responsible for food spoilage in the refrigerator, what can you say? You say they are psychophiles. So let me pause and check the attendance of everyone in this place. I want to know those who are present in this class. At this time, 7:51, I want to see those who are present in the class at this moment, because type yes, present. Type yes, present. 7:51 to 7:52, type yes, present. While I continue the class. Away from temperature classification of bacteria, we look at those that grow based on salinity content. So we have those that are called halophiles. Halophiles require high salt concentration for their growth. We call them halophiles. Because usually salt content prevent microorganisms from growing, but they are those that they require high salt concentration. We call them halophiles. Halophiles are found in Dead Sea. Example is Halobacterium salinarum. And we also have example of Vibrio chloride that can tolerate a wide range of salt concentration because this organism is found in marine environment. Vibrio is a marine bacteria. It can tolerate a wide range of salt concentration to high, to low, to medium. So we call them halotolerant bacteria. Then we move to the pH concentration. pH concentration is the level of acidity or alkalinity of a medium. So we have the acidophile, which thrive efficiently in acidic environment. That is, they can grow in pH medium that is less than 7. They can grow up to 5 pH, 4. We have Theobacillus theoxidans that are found in acid mine drainage. We also have some bacteria that are called lactic acid bacteria. These bacteria as well, they can grow at a lower temperature. But we don't usually call them acidophiles because they can tolerate low temperature, they are not acid lovers. Acid lovers are those that are found in acidic environments. Lactic acid bacteria, they can produce acid in their environment. We also have alkalophiles that grow best in alkaline environment. Example is Bacillus subtilis. These bacteria grow very well in pH above 7, which can usually and normally inhibit the growth of other bacteria. Then we have the antigenic properties. I'm trying to be very fast so that you can finish this class, because I know that some people may be having some network challenges. So we have the antigenic properties. For antigenic properties, it is a way of identifying different bacteria based on antigenic properties that they produce. Now, what am I talking about? Any substance that enters any human body, it's usually called antigen. Why they elicit, they stimulate the presence of substances that fight against disease called antibody. So most of the chemical components of bacterial cells or structures are usually antigenic in nature. Example is capsule, flagella, cell wall, and other anatomical elements of bacteria are source of antigen that cause the formation of antibodies when they get into the cells of humans or animals. So when these substances get into the cells of humans and animals, they usually cause the secretion of antibodies. And these are produced in the serum. We call it anti-serum. They are produced specifically against the particular organisms that are responsible for their stimulation. This will enable these organisms to be identified. It enables us to now classify such bacteria into groups that have the same antigenic composition. Let me come again. Certain organisms, they possess antigenic properties. Examples of these antigenic properties are called capsules. Capsules also form a shield for bacteria. It usually prevents the bacteria from being engulfed by macrophages. Now, when a bacteria is detected in a cell, it usually causes the formation of antibodies. The antibodies that are produced are specific for that bacteria. And when it is specific for that bacteria, then it will now cause the production of these antibodies in some quantity. And it is specific for that type of bacterium. When other bacterium of similar antigenic properties enter the cell, they are easily recognized. So when similar organism or bacterium with such antigenic properties enter the cell of human or animal, the body produces the antibodies. These antibodies is used in classifying these organisms based on their antigenic composition, that is the ability to stimulate the production of antibodies specific against them. So the antigenic composition is often related to the ability to produce disease. And it usually help in determining pathogenic organisms. Some people call every infection that occurs as typhoid. Any small bacterial infection, people believe it is typhoid. Whereas most infection may not be caused by typhoid bacteria, because Salmonella typhi is responsible for typhoid. But most people believe whenever they have any bacterial infection, it's typhoid. So, in a way, Salmonella typhi also has its own antigenic composition that can be detected through serology, which is by antigenic properties of Salmonella typhi. Other antigenic properties that are used in classifying organisms has been explained. We have the cell wall, we have the flagella, we have the capsule, and so on and so forth. So These are used in serotyping microorganisms. It is used in distinguishing strains of bacteria with exceptional virulence properties. And it is of public health concern. Example is Vibrio cholerae, which is a pandemic strain. When such bacteria get into the cell, it is easily detected via serotyping because it will stimulate specific antibody against it. When this antibody are secreted, then we can classify it. Oh, this particular antibody is against cholera. Then that means this bacteria is present, and so on and so forth. We also have E. coli that has different types of strain. We have the enterotoxigenic E. coli, we have the enteroinvasive E. coli, we have the enterohemorrhagic, enteropathogenic E. coli. Now they have the serotypes. They are used in serology in determining the specific strains of E. coli that is causing this type of disease infection. Biochemical characteristics. Most bacteria are classified based on their reaction to series of biochemical tests. So we group them again. This is another scheme. We group them based on their biochemical tests, which are used in a routine laboratory. Example is oxidase test. Oxidase test is used in separating certain bacterium from other ones. Example, Pseudomonas aeruginosa is a typical oxidase positive bacteria. Why? Because it contains cytochrome. Now, in the event where Pseudomonas is marked on a filter paper that consists of oxidase reagents, within few seconds, it will give you a purple color. When you mark Pseudomonas, a culture of Pseudomonas, when you streak it on a filter paper that has been immersed in oxidase reagent, it gives a clear purple color to show that Pseudomonas is oxidase positive. E. coli is oxidase negative, so it will not give you such. And this change is immediately. Vibrio is also positive. So we also have nitrate reduction, which is common among Enterobacteriaceae. We have amino acid degrading enzyme. We also have several ones, such as catalase test. We have urease test. We have so many biochemical tests that are used in differentiating different types of bacteria. In terms of Staphylococcus, we have another biochemical test that is used in differentiating members of the genus Staphylococcus. Coagulase test is a specific test that distinguishes Staphylococcus aureus from other members of the genus Staphylococcus because it possesses ability to make the serum to coagulate. It consists of an enzyme that is coagulase. It makes the serum, which is the medium for the test, to coagulase. And then it is a positive coagulase test in terms of biochemical characteristics. So we have different biochemical characteristics for microorganisms that are used in grouping them in identification scheme. Among the biochemical tests that is very popular is gram staining, which allows a large proportion of bacteria to be classified into two, such as gram positive and gram negative. The topic of gram staining has been overemphasized in your class. So I believe everyone in this class understands what we talk about gram staining. Example of gram-positive bacteria includes Staphylococcus aureus. We have Bacillus anthracis, we have Micrococcus species, Enterococcus faecalis. These are gram-positive bacteria that possess abundance of peptidoglycan in their cell wall. This peptidoglycan allows this organism to retain the gram stain, which is purple in color. Whereas the gram-negative bacteria that has a very thin layer of peptidoglycan do not retain the gram color, which is the purple color. It retains the counter stain of safranin. So under the microscope, the gram-negative bacteria appear in a magenta or red color, while the gram-positive bacteria appear purple. An example of gram-negative bacteria include Proteus, Mirabilis, Escherichia coli, Klebsiella pneumoniae, Vibrio. All the members of Enterobacteriaceae are all gram-negative bacteria. Then we have another mode of scheme of classification, which is the mode of nutrition. For the mode of nutrition, we have organisms that are classified as phototrophs. Typically, we know microorganisms need to carry out certain metabolic activities for them to be able to continue to survive, to replicate, to multiply, to carry out some of their basic functions. They not be with energy. Me that I'm talking energy to speak with you. And phototrophs gain their rather than usual carbon sources. So we have the photolithotrophs and in their carbon sources. They only differ in their chemotrophs. Chemotrophs derive their energy from chemical sources. They don't carry out photosynthesis from chemicals. Example is nitrosomonas. And these organisms are known to obtain their energy from oxidation of inorganic compound. Example is hydrogen sulfide, iron, ammonia, and so on. So they are usually classified as chemotroph because they use chemical for their energy sources. The third category are the autotrophs. They use carbon dioxide as a sole carbon source to prepare their own food. And they are also divided into two types based on energy they utilize to assimilate carbon dioxide, which is photoautotrophs that use light energy and chemoautotrophs that use the chemical energy. So these are schemes of classification of bacteria in differentiating them from one after the other. So we will also look at heterotrophs. Heterotrophs are bacteria that use organic compound as carbon source. They lack the ability to manufacture their own food. They are mostly human pathogens. And the heterotrophs are those that are usually causing disease. Some of them have simple nutritional requirements. And those that require special nutrients for their growth are known as fastidious bacteria, just like I mentioned earlier on. We look at the next topic. We look at two few things on this classification of fungi, and we will call it a day. But we will not call it a day until we have finished this particular aspect. I will stop sharing in 1-2 minutes. I will continue. I want to get your presence in this class. Can I get your presence at 8:09? At 8:09, can I get your presence? From 8:09 to 8:10, I want to know those who are present. 8:09 to 8:10. I want to see your presence. Present. If you are present in this class, type your presence so that I know those who are with me. This class is a large class, and I'm only seeing 117 watching. We are more than this class. Okay, let's move. We will look at this briefly, and we'll call it a day tonight. Fungi are classified into kingdoms. Why do we call it kingdom fungi? Why? Why can't we say genus? Fungi are classified taxonomically as living things with a higher hierarchy apart from other microorganisms such as bacteria. So why the question kingdom? Why are they called kingdom fungi? Why not genus? In classification of fungi into kingdom, it represents their broad level of classification based on fundamental differences in their basic characteristics. That means fungi possess fundamental characteristics that distinguish them from bacterial cells. This is the reason why they are called fungi, kingdom fungi. Oh, how I wish this class is a physical class so I can ask you questions of the next slide before I open the next slide. However, I will move on. Now, what are these basic features that distinguish a fungus from a typical bacteria, and why they are called kingdom fungi and not genus or family? Now, let's look at these basic characteristics. Number one, fungi are eukaryotic. Bacteria are prokaryotic. So eukaryotic cells have two nucleus. They have membrane-bound organelles. That means they have organelles that are in the membrane, like mitochondria, like Golgi bodies. They have compartments for organs, which are similar to plants, similar to animals. So fungi are eukaryotes, they are not prokaryotes. That's number one. Number two is that fungi are heterotrophic. It means they cannot synthesize their own food like plants. They obtain their own nutrients in a saprophytic way. They absorb the dissolved organic matter from their environment. They assimilate it. They secrete enzymes that dissolve their food externally and they absorb it. So they are decomposers. Some of them are like parasites. Some of them are like they are saprophytes, like I said. So they are not, they can't photosynthesize. So they are heterotrophic in nature. Another point is that their cell wall composition is quite different from bacteria, different from plants. Plant has cellulose. Bacteria has peptidoglycan. But fungal cell wall are primarily composed of chitin. Don't forget that. In terms of cell wall, what differentiates bacteria and fungi? Two things, dash and dash. That's a simple question. What are you expect to say? peptidoglycan, and chitin. So in this aspect, fungal cell wall are actually different because their chitin is similar to exoskeleton that are found in insects. So insect belongs to kingdom, and insect cannot be classified as genus. So fungi is having more qualification in terms of this nomenclature. And in growth form, fungi come in variety of shapes and sizes, just like we have different animals, different plants that come in different shape and size. They are not constrained to just one cell. These are one of the reasons why fungi are called kingdom fungi. They have hyphae that are collectively formed in their cell. So these four basic components, being eukaryotic cell, heterotrophic in nature, their cell composition, and their growth form, distinguish them from bacteria and makes them to be classified as kingdom and not genus. Don't forget. In classification of fungi, we have 5 kingdoms. I mean 5 major phyla that were established according to their molecular characterization and mode of reproduction. So the later classification of fungi no longer uses imperfect fungi, which is the Ditromycota. It has been replaced. So we don't have Ditromycota again. For those that have been taught imperfect fungi, it is known now that we don't have anything like imperfect fungi again, because this is used for those fungi that reproduce only asexually. Now, fungi are classified into phyla, which are Chytridiomycota, Zygomycota, Ascomycota, Basidiomycota, and Glomeromycota. In our next class, we will look at this in details. We will look at this in details and we will have more discussion. So today's class, as it is, I don't know, I know this is a live streaming class. We may not be able to ask questions. However, if you have any question, kindly drop it at the comment section after the live streaming. After the live streaming, I will check it. I will respond to you. Then we will have our other class, either physical or online, as the case may be. But for now, we have come to the end of this class. Thank you for those who are present. I will harvest the details and I will respond appropriately. Thank you so much.